Hello, everyone, and welcome to this webinar that's being hosted by LabRoots and sponsored by Infinity Bio. I'm Katy Shaw-Saliba, and I'm the VP of Scientific Development here at Infinity Bio. It's my distinct honor and pleasure to be joined by Dr. Linda Kusner, who is a professor at George Washington University, and Dr. Jessica Lasky-Su, who is an associate professor at Brigham and Women's Hospital and the Harvard Medical School. Today, we'll be talking about how measuring the reactivities of the circulating antibodies through reactome profiling can provide novel insights into human health. Before I turn it over to our speakers, I'll present to you a little bit about Infinity Bio and our novel technology. We'll open up as well for Q&A at the end, but please don't be shy in sharing your questions and answers, your questions as we go. All right. Infinity Bio is the antibody reactome profiling company. Our scientific co-founders are Dr. Ben Larman from Johns Hopkins University and Dr. Steve Elledge from Harvard University, both of whom are giants in the antibody reactome technology space. Our laboratory is located in Baltimore, Maryland, where we operate as a fee-for-service business. We have 9,000 square feet with dedicated laboratory staff, and we are able to process about 2,500 samples per week. Of note, during our Series A closing, we acquired another antibody reactome profiling technology that was produced by a company formerly known as Ceramune, and we've expanded our scientific group with this closing. So you may be wondering what antibody reactome profiling is and what is the antibody reactome. The antibody reactome are all of the possible binding interaction between antibodies and antigens. And so as depicted here in this artistic rendition, what we can see is all of the antibodies present in a person are a ledger of prior and ongoing immune responses. And so they kind of store all of this information, both to foreign and self-antigens. And we believe that this can be very important opportunity for novel discoveries, both in health and disease, as well as serve as a source of therapeutic molecules and diagnostic analytes. How do we read the antibody reactome at scale? Well, we use a novel technology that was developed in Ben's lab at Johns Hopkins by our R&D director, Joel Cradle. And Joel has an incredible webinar on this technology that's on our website. So please feel free to watch that to learn more about it. So this technology is called MIPSA, which is the molecular indexing of proteins by self-assembly. And the way this works is that we take databases of antigens, protein sequences, and we're able to use our MIPSA technology with self-reassembling expansion to generate DNA barcoded antigen libraries. And these can be both full-length proteins and peptides. So each antigen or each protein or peptide is covalently linked through a halo tag to its own unique DNA barcode. So we're able to determine what antigens an antibody in a specimen interacts with through sequencing the DNA barcodes. The workflow of this is shown here. So what we do is we take the antibody source, which is provided by the customer, and we're able to mix this with our MIPSA library in a 96-well plate. We allow the antibody and antigen interactions, and then we're able to capture these through a standard immunoprecipitation, and again, sequence the DNA barcodes. We always compare these then to a mock IP, which contains no antibody source. And our bioinformatics team then is able to translate this into the data is shown here, which are the actual hits or the antigens that the antibody is able to interact with. So what you can see here is each blue bar represents a biological specimen. There, where it says no hits are our mock IPs, and then the green bars are our internal controls. And so you can see the total number of antigens that antibodies in the sample interacted with and see the spread between the different samples, highlighting again that each individual has its own unique antibody reactome profile. We are able to use diverse antibody sources from different biospecimens. And if your favorite biospecimen isn't shown here, we're able to work with our R&D team to accommodate for it. We're also able to use different antibody isotypes depending on what your question of interest is, whether that be looking at IgG or IgE, IgM, or IgA. We have 4 main catalog antigen libraries with non-overlapping barcodes, which means that these can be mixed in a single reaction. The 4 libraries are shown here. VirSite is known, all of the known viruses that infect humans. Our newest library is in VirusSite, and this is the world's largest panel of non-viral and microbial antigens. So this includes pathogenic bacteria, fungi, parasites, as well as a large component of the microbiome. And it also includes allergens from arthropod vectors, food, plants, and environmental allergens. So these two, the VirSite and VirusSite, encompass the external exposome. And then we have the self-exposome, which is our Hugh site. So this is all human proteins as a mixture both of full-length proteins and overlapping peptides. And while I've said we have 4 libraries, the 4th is not shown here, but it's called MuSite, which is the complement to HuSite for mouse models. So these libraries contain defined antigens, but during our Series A, as I mentioned, we also acquired another technology, formerly known as SARA, and we're calling this XSite. So this is where we have bacterial display of random peptides, and this allows us to explore both the dark matter of the antibody reactome as well as the defined antibody reactome, casting the widest net. You'll hear today about applications of this technology, but we just wanted to highlight that there are many different ones covering both the discovery and translational spaces, everything from novel discovery, disease etiology, mechanism of action, and therapeutic targets, all the way through disease stratification and drug response. So without further ado, I'll turn it over to our speakers. And again, please continue to post your questions in the chat so that we can get to them. All right, so Jessica, I will turn it over to you.
Well, thank you. Can you see my screen okay too?
Looks great.
Great. Well, it's wonderful to be here, and I'm really excited to talk about some of the initial work that I've been able to do on the Infinity Bio platform. And really this came out of an interest of really trying to understand how autoantibodies, viral reactivities interact with disease. And, just to lay the foundation, antibodies arise when the immune system mistakenly recognizes self-proteins as foreign, and this can happen in multiple ways: genetic predisposition, namely the HLA region, different environmental triggers, or other stochastic mistakes, quote unquote, are all ways that antibodies can arise. But though I think the one thing that is very clear, it's not really until recently that we could really comprehensively profile autoantibodies and understand how they relate to diseases more broadly. And really, that is what motivated me to do some of this work. And in particular, I wanted to really understand this interrelationship between how viral exposures may relate to autoantibody activities and disease. So in this talk, I'm going to be talking about this interrelationship in multiple steps. And really just focusing on this first relationship is the first thing I did, was to look and see how we could understand the relationship between viral reactivities and autoantibodies. And I'll just put this as the general characterization of what I was trying to do, is really to characterize how specifically herpes viral reactivities relate to autoantibody reactivities. And this is where, in looking at this connection, what we wanted to do, this is really motivated by a lot of work that I'm sure many people are familiar with, where we see very clear associations between past herpes viral exposures and then the incidence or development of disease. And I think one of the classic examples is EBV virus and the development of multiple sclerosis, where through molecular mimicry on the viral antigens that share similar structure to sort of host proteins, then instigate this cross-reactivity with self-antigens. And then that precipitates into the development of diseases like multiple sclerosis. But my question was really, okay, well, we know that in some cases for some diseases, there really hasn't been a broad characterization of how this may happen or the interrelationship between herpes viral exposures and autoantibodies more broadly. And this is a really important question because we know that herpes viruses are established precursors to a lot of different autoimmune and chronic disease outcomes. As I just talked about the example with multiple sclerosis, but multiple other autoimmune outcomes. And this really, motivates this question of, are there underlying mechanisms we can understand here better? And in particular, can we understand if there are specific autoantibody reactivities that are strongly associated with certain herpes virus exposures? So thinking about that, what I did is I went to the Mass General Brigham Biobank, where we have hundreds of thousands of different types of biosamples. And I've done a lot of work in the biobank curating other types of clinical data and omic data and molecular data, as you see here, which is something that is really advantageous as I proceed because we can integrate the data together. But we wanted to add on viral and autoantibody profiling. And so that's when we went to Infinity Bio and we selected a group of samples for both a discovery cohort and a replication cohort. So here you can see we have a discovery cohort of about 12,090 people and a replication cohort of 300. These are just going to be named for here: Mass General Brigham Biobank, MGBB, LLF, and ABC. These are acronyms for how they were ascertained. And then we went and using Infinity Bio, we profiled viral reactivities and human reactivities using VirSite and HuSite. And then we wanted to look at these first interrelated with each other, and then we wanted to understand how these might relate to disease. And in this case, we used all of the herpes viral peptides along with the 4,600 full-length human proteins. Now, the advantage of using something like the Mass General Brigham Biobank is it's connected to over 30 years of electronic medical records. So this inherently makes it so we can look at a broad range of diseases quite well. When we look at these two cohorts, we also see here, if you look at plots C and D, this is the discovery and replication cohort, that we have a really wide range of diseases with pretty good prevalence. And this is because the biobank ascertained individuals through a medical setting. So the levels of different disease diagnoses are higher than one would observe in a typical population. And here are some of the first results we wanted to look at, which is just the overall prevalence of the different herpes virus reactivities, then the discovery cohort and the validation cohort by the type of herpes virus. And we identified here the various herpes viruses. And what you can see is the distribution of prevalence of the peptides is largely the same, whether you have 12,000 or you have 300. But what we can see is that for certain diseases like Epstein-Barr, we have a larger number of peptides that are highly prevalent. Same with what we see for varicella, which is probably a result of vaccine. But again, it's always important to keep in mind this distribution of reactivities. And then not surprisingly, when we look at human reactivities, right, these reactivities are much more rare. And so we really see that the majority of these reactivities are, below about 15%. So in trying to understand this relationship between autoantibodies and herpes viral exposures, the first thing we did was just look at that association. And one of the things we found was some very striking associations between antibody reactivities and specific herpes virus. So in our discovery population here, you see a Manhattan plot. The dashed line is genome-wide significance, and a 10 to the negative 6 genome-wide significance. And you can see the number of autoantibodies that are above that is tremendous. this, right? So suggesting this relationship between herpes virus exposures and autoantibodies is very, very strong and very, very compelling. But of course, what's most compelling are some of these hits that are like 10 to the negative 80. And those are specific autoantibodies that we see, PHLDA1, ZNF550, and P3H4. Now, when we looked in the replication population, what was equally striking was that even though we only had 300 individuals here, these top associations still show up and light up like a Christmas tree, right? And really suggesting that we have good resolution to identify some of these interrelationships between viral peptides and autoantibody reactivities. So when we did this association in total, out across 7 herpes viruses, we identified 37 full-length autoantibodies that replicated across both cohorts. Now, one thing I want to say is as we go from a cohort of 1290 to 300, we have more power in a cohort of 1290 than obviously we do 300. So as different reactivities, either the autoantibody reactivities or peptide reactivities, become more rare, our power is limited. So the biggest limiting factor in actually validating a larger number of these is actually just statistical power. In fact, of those that we actually were able to replicate, 25% of our associations replicated, which is remarkably high. And here is a look at the top in our discovery cohort and at the bottom in our validation cohort of a diamond plot of these replication results. So on the bottom here, you see the different autoantibodies, and then you see the different viral peptides. Now, the lighter the diamond is, it means the stronger the p-value association. So as you see here, we have PHLDA1, which we saw very clearly before. But what I also want to point out is what we can understand when we look across all of these autoantibodies. And in particular, we see CMV seems to have the largest number of autoantibody associations, whereas a lot of the other associations seem to be more unique or more specific to one type of viral peptide, or at least one class, so like the simplexes. Now, over here, we have another way of visualizing this. And what is remarkable is when you look at these two cohorts, the similarity in the associations we see are just tremendous, really just substantiating the validity of these findings. So overall, I think one of the things that really stuck out was this high degree of autoantigens provoked by herpes virus species. And then in particular, of those that we did have statistical power, the clear ability to validate these with remarkably similar results. And so that's one thing we wanted to look at. But another question we have is, if we are going along this pathway of something like how are autoantibodies causing potential diseases, and how are autoantibodies related to the herpes viruses, right? In terms of mechanisms, one of the mechanisms we were interested in is this potentially molecular mimicry. And so this became the next point that we wanted to look at. But before we look at that, just to summarize, up to this point, we reproduced 37 different herpes viral autoantigen associations. Of those, we were able to test 25% of the associations validated. CMV had by far the most viral peptide autoantigen associations, and there were clear autoantibodies that really stood out, in particular PHLDA1, ZNF550, and these associations, were low as p-values of 10 to the negative 80. One of the things that's really important about a lot of these autoantibodies is that the protein itself has very clear associations with disease. So for example, PHLDA1 is known to be a tumor suppressor. It's been associated with prior diseases, including cancers, most importantly, but autoimmune, neurodegenerative, and metabolic diseases. But the autoantibody itself is not something that has been reported. And so that's something I want to highlight that I'm seeing over and over, that many of the proteins we've seen have been associated and reported, but not the actual autoantibody. So the next question was, can these herpes viruses viral peptides actually predict potential antibodies, because it's one thing to be associated, but how well is it actually able to predict those autoantibodies? And so we looked at this across just the 37 replicated associations. And I'll go through this by each herpes virus. This is the test and valid test training and testing sets for HSV1 viral peptides being able to predict PH-LD1. And in the training, it was able to predict it with, if you look at just the red line here, with 88% accuracy. What's amazing is in the test set, it was able to predict this at 97.3% accuracy. And I'm actually realizing these plots should be switched. But what's really notable, the testing set is more accurate than the training set. In this case, too, when we looked at this in AlphaFold, we found quite good sequence homology between the particular peptides that were predictive of PH-LDA1, suggesting that is consistent with molecular mimicry. Now, we look at the other associations. This is association between Epstein-Barr virus and P3H4. Again, we see the same thing where we have, a training set, but then the testing set has even higher accuracy. And we did this also with CMV. We actually had 3 different autoantibodies that were associated or that were accurately predicted with over 90% accuracy. For CMV. What was really interesting about this, though, is all 3 of these autoantibodies, the main prediction in that association was one particular viral peptide. So what can we conclude here? Not only can we see that the relationship between herpes viral reactivities and autoantibodies is remarkably strong and reproducible, we now see that there is very good ability to predict with high degrees of accuracy some of these reactivities with those viral peptides. And in some cases, as in the case with PH-LDA, we see quite good sequence homology, consistent molecular mimicry. And in other cases, it looks like there might be one viral peptide in particular that is really driving a lot of this prediction. And finally, while I only showed the results that validated with over 90% accuracy, I want to highlight that of those 37, many of the autoantibodies were able to predict with a pretty good accuracy of 70 to 90% as well. Okay, so we've looked at viral exposures and autoantigens, and now I want to add the last part in, which is, can we understand how these interrelate with disease? And so the aim of the second part of the study is really to characterize a relationship between herpes viral peptides, their associated autoantigens, and disease. And as I mentioned earlier, one of the unique advantages we have in the biobank is that we have a large number of disease diagnoses through the EHR data, and we have 30 years of data. And my group has curated, it's probably close to 100 different disease diagnoses now, and we're able to look at those diseases with respect to the antibody profiling data. And so we can look at both incidence and prevalent cases in different analyses to try to understand this relationship. So in this next part of the study, what did we do? Well, we really wanted to look at 3 different relationships: the relationship between viral peptides and autoantigens, which we already did. And then we want to look at the relationship between the herpes viral peptides and disease, and the autoantigens and disease. And so to do that, what we did was we did all of the associations, and then we visualized it in tripartite networks, which show all of those interrelationships. So here is an example of one of these networks where you see viral peptide nodes are in green, disease nodes are in pink or red, and autoantigens are in blue. And so here you see these are all the viral peptide reactivities and their relationships to disease in green, and their relationship to autoantibodies in blue. And what you can see here is one of the things that's quite striking is the autoantibodies that we see associated with the different herpes virus are also associated with several of the diseases. Now, if we see the diseases that are in red, the nodes that are in red, that means both an autoantibody and a viral peptide are associated with those diseases. And here's a really good example where we see both PHLDA and PPA2 are acting not only as associated with the viral peptides, but they are also acting as disease hubs, meaning that they're associated with a range of diseases. And if we actually look at the top 20 associations, disease associations by viral peptides in this bar graph, there's several things we can see that are striking. Number one is, the strong relationship we see between a lot of these diseases and viral peptides. Many relationships where odds ratios are greater than 10 in these associations. And if we look at these top lists, there's certain hallmark characteristics that we see coming up, which is cancers, autoimmune diseases, neurodegenerative diseases. And in particular, in this case, you can highlight, that some of the very top associations are cancers here. So I just want to highlight in this, well, the top associations are cancers, and that isn't necessarily where we see the peptide association, the autoantigen associations. When we look at this other range of diseases, we see all of the pink associations, which are the autoantibody associations, really adding a second level of validation that these viral-associated autoantibodies are also associated with same diseases. Now, we can actually do this with each of the different herpes viruses. Here is HSV2, where we see quite a similar overall scenario. But in this case, there's actually, looks like there may be less association specifically between the autoantibody and the viral peptides than we did in this last one. Again, if you look here, you'll see there's a lot more blue there, right? So what we do see again, though, is the same idea that a lot of the viral-associated autoantibodies are also strongly associated with similar diseases. And over here, you can see again, some of the very top diseases like giant cell arteritis has an antibody and viral peptide associations. When we look across the range of diseases, we actually see a relatively similar types of disease domains, yet the specifics of each of the particular diseases are quite distinct. So here we see some infectious diseases more prominently, and we see Alzheimer's here more prominently. So a little bit different profile of the actual associations. And again, just validating this idea that we can use both viral peptides and autoantigens associated with them to further validate some of these diseases. So going on to the EBV network, this network had fewer autoantibody associations overall. Yet those autoantibodies still acted as hubs. But because of that, when you look at the overall plot of viral peptide disease associations and autoantibodies, we see fewer autoantibodies here. And the range of that effect is quite a bit smaller. Now, one of the things I want to highlight here is this isn't actually too surprising because a lot of the EBV peptides were highly prevalent or very rare. And so of all the different herpes viruses we looked at, we have the most limited ability and most limitations in terms of statistical power to actually identify these associations. Yet nevertheless, we still are able to validate associations like multiple sclerosis, and we see one really clear disease hub, which is CD320. Finally, our last association here is with CMV. And here what you see is it is just completely dark because there are so many associations between viral peptides and diseases, viral peptides and autoantibodies and disease. So overall, you don't want to get CMV because your increased risk of all diseases seems to be much more elevated. And you know, this is so pronounced here and so clear, but not only is that clear, when we look at the actual effect of these associations, some of these association effects are huge. So, you know, an exposure to CMV, you have viral peptides associated with prostate cancer with odds ratios greater than 42, right? So this is a huge jump in terms of risk. We see the same general disease profiles, but if you look over here at the bar plot, the number of autoantibodies that we detect here throughout all of the diseases is much more pronounced. And the effect sizes of these are much more pronounced. Interestingly, we also note that some of the autoantibodies that we see, we know are well-known players in autoimmune diseases. And one of those is TRIM21, which is known to be associated with a lot of different autoimmune conditions. So in this case, what was actually remarkable is of the viral disease associations, the autoantibodies were able to replicate nearly 60% of those, which is really sort of validating this as a potential mechanism, but also of actual etiology, but also just remarkable similarity between those. So in summary, when we look at this combination of herpes virus networks with disease, there are certain things that are very clear. Number one, you know, the disease associations not only are super pronounced with viral peptides, but they're specific autoantigen hubs that seem to be disease hubs where a lot of the different associations are being driven through as well, or a subset of those. And we can actually classify sort of the overall viral peptide layer association with disease and how that is distinguished from the autoantibody layer of disease, which in some cases is very similar, as in the case of CMV, but in other cases may have a slightly more distinct profile. When we look at this too, I'll point out that while for CMV, nearly 60% of the viral peptide disease associations validated with the autoantibodies, for EBV, it was much less. And really this is driven by the fact that we had less overall number of autoantibodies we detected associated with EBV to begin with. So to summarize, we've identified 37 reproducible herpes virus autoantigen associations. Of those that we've been able to replicate, we had a remarkably high replication rate. See really robust associations for a subset of these, PHLDA1 and ZNF550. Importantly, of that replicated, we also see that viral peptides are remarkably good and at predicting some of these with high degrees of accuracy. When we also looked at and further characterized the disease footprint really expanded on what is currently understood across the herpes viral species. While it validated current results, there's a lot of ways it expanded and made findings more specific, as in identifying particular associations with types of cancer rather than just cancer overall, more broad associations with autoimmune outcomes, et And when we look at this, CMV really stands out as having the largest impact on both autoantibodies and on disease multimorbidity. So, you know, where are we going from here? So this is a really exciting start of what we're doing. The next steps are really going to be looking at autoantibodies overall, right? We only looked at 37 autoantibodies, but we're going to be looking at them overall over those 70 plus diseases. And we're also going to be looking at them in the context of biological aging as well. And to give a little shout out hint of what's coming, one of the things we see that's extremely interesting is a really pronounced sexual dimorphism in terms of associations that are sex specific with females and males. We see very pronounced that there is a protective group of autoantibodies associated with reducing aging particularly in women, that is not existent in men. And so these are some things we're looking into. It's super interesting, and I'm really excited to keep on looking further. So with that, I'll just want to thank the people who contributed with this. The whole group is right here.
Well, thank you so much, Dr. Lasky-Su. So we're collecting the questions, and we'll go ahead and answer them all at the end. But let's go ahead and we'll now have Dr. Kusner present her study.
Well, thank you for the opportunity to talk about thymoma-associated myasthenia gravis and the occurrence of autoantibodies. I just want to know that there's no conflicts in the disclosure related to the subject of this presentation. So to begin to discuss this, the thymus. The thymus is a site of central tolerance where T cells go to for maturation. The immature progenitor cells come in as double negatives. They go through a process where they become double positives. An interaction with MHC class 1 and 2 produces a positive selection where the single positive CD4, CD8 migrate into the medulla where they interact with self-proteins. This self-protein expression is modulated by air expression. The medulla also contains myeloid cells that contain high levels of skeletal muscle proteins. So this interaction with self creates a negative selection where the T cells binding to self will cause, to high level, will cause apoptosis, or they can mature into regulatory T cells. Those that do not bind high affinity will then go be secreted as CD4, CD8 mature T cells. The thymus involutes during aging and is replaced by fatty tissue. However, the maturation of T cells continue. So in thymoma, it's a rare neoplasm of the epithelial cells. This is a very slow growing cancer. Its classification, as seen on the right, is based on the morphology of the epithelial cells and the proportion of the associated immature T cells. A through B it's more considered a similar type thymoma, where C is definitely distinct as being a carcinoma. Most of these patients have deficient air expression in the tumor cells, and patients also have the low expression of MHC class 2, resulting in a decrease in Tregs, and they also produce autoantibodies to the type 1 interferons, and it's associated with a high level of autoimmune disorders. Now this next slide is, this is a listing, a partial listing of the autoimmune disorders associated with thymoma. There are other systems involved, not just the neuromuscular, but this highlights the fact that 30 to 44% of these thymoma patients have a prevalence to myasthenia gravis, compared to other autoimmune disorders where the percentages hover around 1%. So what is myasthenia gravis? It's a rare autoimmune mediated disease caused by autoantibodies, and they target the acetylcholine receptor as well as the muscle specific kinase and LRP4. The majority of patients have antibodies to the acetylcholine receptor, and due to the interaction of the acetylcholine receptor, it causes an interference between the nerve and the muscle interaction, which is seen with weakness and fatigue in the muscle groups. Often occurs within the eye muscle, so you would have diplopia or double vision, and the disease can progress to a generalized state involving the extremity weakness. If it involves the respiratory muscles, the patient can undergo a myasthenic crisis. So in thymoma-associated MG, as I mentioned, it's about 30% of these thymoma patients, although the MG patients, only about 10 to 15% will have a thymoma. These autoantibodies include the acetylcholine receptor, titin, ryanodine receptor, as well as the type 1 interferons. So our hypothesis going forward is that the thymoma-associated myasthenia gravis patients would have a greater diversity of circulating autoantibodies compared with a non-autoimmune controls. So we looked at the Infinity Bio MIPSA technique in order to determine these autoantibodies. Our subjects, we looked at controls through our George Washington University Biobank, and we aged and sex matched them to the thymoma patients. For our thymoma myasthenia gravis patients, we went to the Indiana University that has a large center biobank of thymoma cases, where we obtained 60% of them. Most of them had known acetylcholine receptor titers, although those that were not did not have a titer associated with them, diagnosis was made by clinical symptoms, and they varied in terms of their HU classification. So going forward, we just assessed the entire population of samples and showed that close to 5,000 proteins were autoreactive with over 6,000 peptides. There was a wide range from in our thymoma-associated MG patients. There was 26 to 415 of the proteins, where our controls had 47 to 623. We had 2 controls that showed high levels of reactivity. However, with that, neither of the peptides or the proteins showed differences. There was a higher level of unique autoantibodies in the thymoma-associated myasthenia gravis subjects compared with the controls, with greater than 2,000 proteins and close to 5,000 peptides found to be autoreactive. So questions we hope to answer with this is how much commonality is there among the thymoma patients, and what are these autoantibodies, and can they predict the occurrence of the thymoma-associated myasthenia gravis? So we clustered these subjects based on their hits on the proteins. And you can see on the right-hand side, we have the clustering of controls. And then in the center, we have a clear distinction of the thymoma-associated myasthenic group. And that band in the center are the autoantibodies directed towards the interferons. This did not pick up, however, the acetylcholine receptor autoantibodies. So we looked closer at these autoantibodies to the interferons. Interferon alpha has 13 distinct subtypes, and we picked up about 10 of them in 23 of our 40 samples. 14 of those 23 samples also showed autoreactivity to the interferon alpha peptides, and there was no relationship between the reactivity to interferon alpha and clinical characteristics. Of the 23, 3 subjects also had Good syndrome and 2 had Isaac's syndrome. So about the interferons, they are produced to a response of invading viruses and bacteria that trigger the TLRs to produce interferon. So in an autocrine paracrine fashion, they actually regulate an inflammatory response. So in a study back in 2012, they looked at several subtypes of myasthenia gravis, finding that the thymoma-associated myasthenia gravis had higher potentials for the expression of antibodies to alpha-2 and alpha-8. The functions of these antibodies to the interferons were considered to be neutralizing. Therefore, these patients that had expression of these autoantibodies seemed to do better because of its ability to suppress that inflammatory response. And then going further, we looked at these 23 patients for other potential autoantibodies, and we found that there's a reactivity to TRIM46 and P2X4 receptor. Now, TRIM46 is upregulated in cancers, and there has been documented literature on the expression of autoantibodies in the paraneoplastic central nervous system disorders. P2X4 receptor is a purinergic receptor. It's found in multiple immune cells, and it's known to be involved in inflammation and immunity, although there is no literature that suggests autoreactive antibodies to P2X4. So with that, my conclusion is that there's a number of reactivities to proteins and peptides that were similar in the thymoma-associated myasthenia group compared to controls, although the thymoma-associated group demonstrated more unique autoantibodies. 23 the samples demonstrated autoreactivity to the interferon alphas, where those 20 also contained antibodies to the interferon, to the TRIM46, as well as the P2X4 receptor. And with that, I'd like to acknowledge the group at George Washington University, as well as Indiana University, Dr. Lauer and Manor, who are experts in thymoma. We are going to discuss the interferon alpha expression with Dr. Kusner, University of Tartu, who's an expert in the field and has actually published several articles related to the expression in thymoma. And then Stephen Manser has an expertise in the P2X4 receptor, and we're working with him in terms of developing an ELISA base to verify that. And with that, thank you very much.
All right, wonderful. Thank you so much to both of our speakers. These were excellent presentations, and I learned a ton from them. We do have a number of questions in the question and answer chat, so please continue to put your questions there and we'll try to get through them. I think we'll start out with the questions for our speakers. So this first one is for Dr. Lasky-Su. For the tripartite network analyses, were you able to identify associations for cancers that have herpesviruses known as the etiological factor?
Yeah, and I did actually try to do it where I responded in the chat, and so I sent a message. So it actually, and I said, actually, we actually see a huge number of cancers, and I was going through the list. So we see, for some reason, I don't have Hodgkin's lymphoma. I don't have annotation non-Hodgkin's. We see non-Hodgkin's and leukemia very strongly. Then the other ones are bladder, lung, uterine, colon, liver, breast, prostate. So we see almost all of them associated with EBV.
Fantastic, thank you. For Dr. Kusner, we have one that says, why were AChR antibodies not identified?
The acetylcholine receptor is quite a complex protein. It's very difficult to actually assay, so I was not really too surprised. The radioimmune assay is the only known ability to actually identify this, and it's done through the Mayo Clinic. It's a large, complex protein.
Thank you. Yeah, and I think that gets at, we have a question about transmembrane proteins. So that is a little bit of one of the limitations with the assay is that it's really hard sometimes with these very large multi-pass proteins for any of the antibody reactome profiling technologies. We do have, obviously, in our human site panel, we do have full-length proteins. So if there is a conformational epitope, we'd be able to pick it up there. But again, the large transmembrane proteins are a little bit difficult in the platform. All right. And then Dr. Lasky-Su, I'm sorry, I can't see the answer. So I'm sorry if you've answered this one in the chat, but we're all viral peptide and autoantibody disease associations in the positive direction or any for protective antibody responses?
I also did, and I don't even know if I did it correctly, but I did respond. But so that is actually really interesting. And I actually gave an example of some of the distributions that we see. So the answer is we absolutely see different autoantibodies that are protective. And to me, that is a super interesting area. And I can actually, I might be able to pull this up because this is something that we're working on now, but you can see here. So what's interesting with this is, number one, if you look at the overall distribution for most diseases, if you look at a volcano plot, you're going to see that it is right skewed. So there's a lot more that are positively associated with disease. Some cases, though, we see some where there are some that are protective against disease. So we actually looked at this really carefully with like the framework of looking at biological aging phenotypes, because we have different aging clocks in our biobank. And what we actually see is, particularly in women, there's a group of autoantibodies that is strongly associated with reduced biological aging. And then when we look at that, we see those same autoantibodies are associated with a protective effect across a lot of diseases. So I actually think this is a pretty underexplored area that there could be some interesting findings in terms of just understanding, potential protective mechanisms of some of the autoantibodies.
That's fascinating. We really look forward to learning more on that as well. For Dr. Kusner, I was wondering about what you kind of consider your next steps from your project and your major goals going forward.
So we like to verify all the autoreactive proteins that we found to verify that, first of all, are they in the thymoma-associated MG group, but then to go further to assess them, their presence specifically in that group, especially with the TRIM46 and the P2X4 receptor. We also would like to look at the function of these autoantibodies, as mentioned. Are they protective? Are they potentially could be activating the P2X4 receptor? It could bind to the ATP site and actually activate that channel, creating this spontaneous increase in calcium in the immune cell and cause activity. So it's an interesting steps forward.
Definitely. Yeah, we look forward to that as well. Wonderful. All right, Dr. Lasky-Su, I'm hoping you haven't answered this one yet, but have you looked at the mechanism by which the autoantibodies cause disease, kind of following up with next steps for Dr. Kusner?
No, and that's exactly where we want to go. What I've realized is when you look at this and you're finding a lot of these autoantibody associations, that becomes like the very next logical step. And we haven't done that, that's really where we want to go. But the one thing I will say is there are several cases where what you see as the autoantibody is really logical. So the potential function could be very clear. And I'll give you an example of that. We see that an autoantibody to a gene that causes early onset Parkinson's is also super strongly associated with Parkinson's overall. So in a case like that, when you know that the gene is a gene that's a functional gene, and if that gene, that gene has loss of function and causes Parkinson's, you already sort of have a good idea of the function. So we are definitely seeing some cases like that where it seems like there's an obvious way to sort of target looking at function. but we haven't yet, but I'm looking forward to it.
Wonderful. All right, we have a few questions kind of around the technology. One of the questions is about antibodies that are polyreactive. So kind of, do we see antibodies that pick up different parts of the protein? Maybe Dr. Lasky-Su or Dr. Kusner, if you saw that with your PROMAX results.
That is one thing we saw definitely in terms of like when we look at the associations, right? That there's certain peptides that seem to be more strongly associated, but then also, for the viral autoantibody, right, are the same ones that you can sort of see sequence homology. So sort of what we've sort of seen there that I expect is there's going to be specific peptides that we see that are the driving forces.
Great, thank you. And just one clarification, I think that the question was more on polyclonal. So that was
Sorry.
No, that was my fault as well. Oh, great, thank you. All right, perfect. All right, let's see. Going back, there were a couple questions we had just about our libraries on the antigens, human or pathogen. So our VirSite and our EnviroSite libraries, those are all non-self, whereas our HuSite library is all the autoantibodies. So kind of broad range of antigens. Let's see here. All right, so for Dr. Lasky-Su, how do you deal with homology between herpes viruses when assigning autoantibodies to VirSite signal? Or how does the platform deal with shared sequences between related viruses? That's a great question.
And I would defer that to you because we do rely on your annotation.
That's right. So we do have some bioinformatics ways we look at this. We have some multiple sequence alignments that are done to conserve as much of the diversity as possible. There's kind of different ways, I guess, in the analysis. You can look at conserved proteins versus ones that are not as conserved. So thinking about in SARS-CoV-2, the nucleocapsid protein versus reactivity to the spike protein. We can also look at the sequence at the peptide levels as well to kind of determine that. But that is always an issue, I think, in using different serological methods. And it also kind of depends on the annotation within the library. All right. And then we have a question about sharing papers. So I think both of our speakers are probably preparing their publications. So hopefully we will be able to share those soon. Going through peer review processes, we all know, has its own speed. So, all right. Are there any other last minute burning questions that people may have? All right, and I know that we did have a few kind of very in-depth technical questions. We're more than happy to reach out and discuss more with you, kind of about the volumes, costs, validation, those questions as well. And then we do have a question about the presentation. Yes, it will be recorded, and we also will post it on our website. And our previous webinar on the MIPSA technology, more in depth, that Joel presented is there as well. And you should be getting an email alert about the recording. All right, well, if there's no other burning questions, do feel free to reach out to us. And I just want to thank Dr. Kusner and Dr. Lasky-Su again for their time and their wonderful presentations. Thanks to LabRoots for hosting us, and on behalf of our entire Infinity Bio team. So thank you, everyone.